Sure.
Jim Kublin is an acclaimed global health expert who works on the development of vaccines and treatments for life-threatening infectious diseases, including HIV, tuberculosis, malaria, and COVID-19. As executive director of the Fred Hutch, headquartered in HIV Vaccine Trials Network, he coordinates its massive international efforts to test vaccine candidates. He now plays a similar role for the COVID-19 Prevention Trials Network. Since July of 2020, has been managing large-scale testing of vaccines against SARS-CoV-2. Dr. Kublin has carried out extensive research in malaria and HIV in South America, Southeast Asia, and Africa. In his role as medical director of the Fred Hutch affiliated Seattle Malarial Clinical Trials Center, he continues to oversee studies on new malarial drugs and vaccines. At Fred Hutch, his team in the Kublin lab conducts preclinical studies in the effects of the microbe on vaccines, microbes that live naturally in the gut of humans, and focusing on how responses of the immune system are changed by the interactions with specific microbes that live naturally in the gut of humans and animals. Dr. Kublin completed his bachelor's and MD at Georgetown University and received his MPH and completed a residency in preventative medicine at Johns Hopkins University. Dr. Kublin continues his work in vaccine development and molecular epidemiology while attending the University of Maryland School of Medicine for his fellowship in vaccinology and the Center for Vaccine Development. Thank you so much for being here today, Dr. Kublin.
Sure. Thanks, Jenny, for the invitation. I'll have to shorten that bio sketch, it seems. Well, I really appreciate your flexibility and interest in, of course, this important topic. Let's see, I'd just like to get rid of my, I'm looking.
Well, I just wanted to chime in that I think it's so important that pharmacists really well understand the vaccines and the vaccine development process so that they can make strong recommendations to their patients, and that it's so critical that they have that confidence and understanding there and can really translate that out to their communities and their patients. And just on a personal note, that was why I thought this was just such an important topic to bring and discussion to have today. So thank you.
Sure. No, and thanks again for the invitation. I think you're exactly right. It's so critical to have some familiarity and awareness with how these vaccines are coming to market. We're quite confident that they will come to market, and I'll mention some of these reasons why we think there is a high probability of success for an effective COVID-19 preventive vaccine. So this is the outline of the talk today. I'll just be speaking very briefly about the pandemic and the virus, some of the epidemiology and health disparities that exist in our country and what we're learning about that and how to take that into consideration for these clinical trials, the different vaccine platforms that are being evaluated, and then some background on the CoVPN, or the COVID-19 Prevention Network, that is contributing to the design and the implementation of these clinical trials worldwide. I'll speak a little bit about the pipeline and the operations and some of the clinical monitoring challenges, and close with a brief description of our volunteer screening registry and what we've learned. And what we've learned is an amazing amount over the past year. Not even a year, of course, since this emerged a year ago next month in December in China, followed by a more global appreciation of that the following month in January, and then the sequencing of the virus in mid-January that really set the stage for a lot of this vaccine work. And it was really in that short period of time between December and mid to late January that a tremendous amount about the virus was learned and applied to the most modern techniques, as well as techniques that have existed for centuries with regard to vaccination programs. So this is the SARS-CoV-2 bad news wrapped in a protein. The red spikes on the surface of this cartoon of the virus are the spike proteins, and these are the very immunodominant proteins that the immune system responds to, and that also appears to be protective. Once one does experience infection, of course, most people are resolve that infection very well, and then proceed to be immune against subsequent infection, at least for a period of time. And we're not really conclusive regarding the durability of these protective responses yet. There is evidence that people have been reinfected after these antibodies wane. So we'll be very interested to look at the durability of vaccine-induced antibody responses, and thus the durability of vaccine protection against this spike protein. Now, the coronaviruses are a grouping of viruses that have existed probably, for hundreds of years. There are 4 non-SARS-like human coronaviruses that circulate seasonally, generally causing mild respiratory illness. And the seasonal activity around these viruses is quite pronounced. And there's some concern then that SARS-CoV-2 could also result in a more seasonal influenza-like circulating upper respiratory infection and systemic immune-mediated disease. This is a snapshot of the Johns Hopkins University Coronavirus Resource Center yesterday, with now almost 50 million cases worldwide, over 1.2 million deaths worldwide. And of course, we've seen the terrible impact it's had domestically, despite some of the concerted efforts with over 230,000 deaths in our country. And on the lower right, we see the daily cases and what we're all aware of being increases in these daily counts and cases worldwide, especially over the past month. So there are a lot of social and structural factors that are impacting COVID risks. Their minority populations are overrepresented in these essential service industries, which result in increased exposure. Low-wage jobs often do not provide some of the foundational protections and availability of care. There's also residential segregation that is founded on some of the race ethnicity demographics across the country, and also increased levels of chronic stress among some subgroups of the population. This was a remarkable publication in the New York Times with accompanied references highlighting the disparity in the cases per 10,000 people across different race ethnicities. And this persists throughout the epidemic nationally. And a very striking and devastating hospitalization rate that we see here, particularly impacting American Indian and Alaska Native communities, non-Hispanic, Black, and Hispanic or Latinx populations, compared to general Caucasians or non-Hispanic, Asian, or Pacific Islanders. In addition to that, we see a geospatial distribution of increased risk with regard to race ethnicity, predominantly overlaid with the risk of COVID-19 as a result of pre-existing medical conditions. So in Mississippi and Alabama, areas known as the Black Belt in those states, in Arizona with native high-density residential locations, even though they're located in very large, sparsely populated geographies, the residential densities in these communities is very high, as examples. And then we've seen not just case rates, hospitalization rates, but fatalities among minority populations disproportionate to those of the general population. For example, representing in Louisiana, Black people are 33% of the population, but reflected 70% of fatalities. And we see similar disproportionate impacts compared to the general population prevalence across a number of these states. And then, of course, we're also very aware of how devastating COVID-19 infection and disease is in the elderly. And this is a decade stratification for age and cases, and see a similar disproportionate impact on COVID-19 death rates among the elderly, and particularly among middle-aged individuals. If you look at the 35 to 44-year-olds, for example, on this figure on the right, the ratio of blacks to white who died is almost is 10 to 1 for blacks, about 8 to 1 for Hispanics in that age strata of 35 to 44 years old. That ratio or delta between death rates among these race ethnicities decreases as these decades later in life are reflected here. So these are all foundations to understanding the epidemiology of SARS-CoV-2 infection within the US, as well as the resulting COVID-19 disease. And so we've given our responsibility to conduct these COVID-19 clinical trials, have really approached this with a concerted community engagement effort to ensure that we do our best to enroll these race ethnicities that are disproportionately impacted by the epidemic into the clinical trials themselves. But that is a formidable challenge, nonetheless, one that we think is critical for us to undertake. So some of the implications of this are when we're actually conducting the clinical trials themselves. This schema on the top provides you a cartoon as to the conduct of an efficacy trial. It starts, we spend 2 to 3 months during which time we're enrolling, recruiting individuals into the study, enrolling them, vaccinating, and then conducting follow-up. Some of the vaccine trials require 2 doses of the vaccine from 3 to 4 weeks apart. Some of them require only 1 dose of a vaccine. And then we follow them. These are endpoint-driven studies. And that means that essentially we need a minimum threshold number of the primary endpoints. And for most of these efficacy trials, it's not just symptomatic disease, but some level of moderate to severe disease. And we've also been in a lot of discussions about some minimal endpoint of severe disease so that we can be confident we're not just preventing transmission, or we're not just preventing mild disease, but indeed we're having an impact on severe disease. So when we look at these schemas and we look at the recruitment strategies by these different groups who are focused on conducting these trials, we're asking ourselves what representation of populations is important for these primary analyses, for the final analyses, and how do we best accelerate the recruitment that must be necessary to increase the representation of these subpopulations. These are some of the questions that we ask while preparing for the studies and deploying them and moving them forward. And this is a nice summary and way forward by colleagues Katie Stevenson and Dr. Basola at the Brigham in Boston. And they've recognized this and published on it earlier this year, and how best to engage these communities. This engagement efforts are critical to ensure the communities are well informed and prepared, we have meaningful relationships, and that there's trust necessary for when we do have efficacious vaccines, we can deploy them in these populations with the confidence and trust that's necessary to overcome some of the vaccine hesitancy that we know exists in many of these communities across the country. So moving on to the actual vaccine designs, this is just a cartoon schema that we originally developed for our HIV vaccine platform, but adapted it and modified it to the SARS-CoV-2. And in moving clockwise at around 3 o'clock, you see Novavax and Sanofi. These are the recombinant nanoparticle protein vaccines that are combined with an adjuvant. It's the more standard, typical way that most vaccines have been developed. But the manufacturing and purification process for these vaccines is oftentimes quite timely. And that is, it takes a long time to get the final product released. Whereas if we continue clockwise in this circle, we encounter the Moderna and Pfizer nucleic acid vaccine platforms. And these were the two vaccine strategies that were the first ones to enter into phase 1 clinical trials. And that's because they're constructed with only the necessary information being the sequence of the target immunogen from the pathogen of interest. So in January, when the SARS-CoV-2 virus sequence was made available, Moderna and Pfizer both jumped on it, included that sequence in some of their manufacturing, and developed prototypes that went into clinical trials within 90 days after that initial sequencing of the virus. An unprecedented really time frame from sequence to first-in-human clinical trials for any vaccine in the history of vaccinology. And then next, the last two here are AstraZeneca and Janssen. They're relying on viral vectors to carry a piece of this spike protein in the genetic material of the virus particle, the virus vector, and it's delivered. And we've tested the Janssen Ad26, adenovirus 26, extensively for HIV vaccines. AstraZeneca built their platform on the Oxford University chimp adenovirus, both of them looking promising with regard to safety and immunogenicity, and are both conducting phase 3 clinical trials now in the US. Now, this is just a more molecular and structural snapshot of this SARS-CoV-2 spike protein, which is, as I've mentioned, the vaccine target. This is illustrated on the left with a coronavirus. On the right is the cartoon, the structure of the spike protein, and modifications to the protein were made based on some of the expertise and work that has been done at NIH with stabilizing the original SARS-1 virus, as well as the MERS virus, the Middle Eastern Respiratory Syndrome virus, both of which are also in this coronavirus family. And so with that background structural work that they applied to the spike protein from these other coronaviruses, they were able to rapidly develop and stabilize the SARS-CoV-2 spike protein. And each of these three platforms, I mentioned the nucleic acids by Moderna and Pfizer, the adenoviral vectors by AstraZeneca and Janssen, and the protein and adjuvant approach that's taken by Sanofi and Novavax. Each of these are eliciting good antibody responses. Typically, the protein and adjuvant elicit the highest levels of antibody. All three of them are inducing good CD4 responses, and it's really the adenoviral vectors that have a very good strategy to induce CD8, typical kind of T cell killer cells that may also be quite helpful in contributing to controlling the spread of the infection. So I mentioned the optimism about developing a SARS-CoV-2 vaccine. We have seen that immunity develops from infection, although there are some questions about the durability of protection. Humans cure the infection over 95% of the time. That certainly is not the case with HIV, for example. And in contrast to HIV, there's no latent infection. So once an individual is cleared of the virus, there is not a latent infection there to resurface. Unfortunately, the infection does produce this pro-inflammatory dysregulated immune response, but we also think that that's an advantage, not just for therapeutic purposes, but for the vaccines themselves, being likely to moderate and ameliorate the resulting inflammatory reaction that's causing much of the severe disease in individuals who may be predisposed, because oftentimes a result of these pre-existing conditions. We know the target for the infection of epithelial and endothelial cells, and that spike protein attachment to the ACE2 receptor is obviously reinforces the target itself, the spike protein. Fortunately, there's little variation within that protein, in contrast to HIV, and it's less variable even than flu. And then there are preclinical non-human primate models that have displayed protection in SARS-CoV-2 challenge models with these very vaccines that are moving into the clinic. So it was about 6 months ago when the thoughts and discussions were had with Dr. Fauci with regard to the leveraging the tremendous infrastructure that the NIH has invested and built for our HIV vaccine trials network. And these are just some of the hundreds of partners that we're working with around the world to implement these clinical trials. A tremendous community engagement effort with many very local community-based organizations, including national regulatory agencies, both within the US and in many other countries outside. So the approach was really to define how best to strengthen this platform that was established and built for HIV vaccines and apply it to the SARS-CoV-2 pandemic. So we thought that, of course, randomized controlled vaccine efficacy trials were the best way to provide rigorous evidence on the safety, immunogenicity, and ultimately the efficacy of these vaccines. And in parallel, we would conduct multiple clinical trials of multiple platforms. So the CoVPN was established, and it brought together a number of different networks. I mentioned the HVTN. There was a recently established infectious disease clinical research consortium, the HIV Prevention Trials Network, and the AIDS Clinical Trials Group, all of whom came together in this concerted effort to focus on both vaccine efficacy trials as well as monoclonal antibody prevention trials. We also realized that the traditional paradigm of moving consecutively with pauses in between each of these phases was not going to deliver to us an expedited assessment of the safety, immunogenicity, and efficacy of these vaccines, but rather by stocking some of them on top of each other, we could accelerate this without compromising some of the key assessments of safety, immunogenicity, and efficacy. So it really came down to recognizing that a global effort, global cooperation, and transparency were necessary to maximize the speed, veracity, and decision-making required to deliver the advances to the global population in a timely fashion. And thus, the CoVPN Operations Center, based here at the Fred Hutch, on the foundation of the HIV Vaccine Trials Network, that has been here now for over 2 decades. In early 2020, we had 52 sites in the US and about another 56 sites in sub-Saharan Africa, about 100 sites worldwide. These were predominantly academically based, and we consider ourselves an academically based contract research organization with particular expertise that is investigator driven and scientifically driven. Larry Corey and I leading the Operations Center, Peter Gilbert, our fantastic computational statistician, leading the Statistical Data Management Center, and Julie McElrath leading the world-class immunology and virology labs. So recognizing that optimally we would focus on the development of multiple vaccine platforms, recognizing that not always do all platforms make it over the final line to safety and efficacy after these late phase 3 clinical trials. We also recognize that the manufacturing challenges of any single platform was not sufficient for national, let alone global production of sufficient vaccine. So we focused on multiple platforms, manufacturing scalability, a coordinated effort with the US government and these global vaccine manufacturing companies, and this coordination led out of Fred Hutch, requiring a global effort. And it's this global effort, here you see the footprint of our clinical trial sites as of early August. We've been moving so fast, we haven't been able to update our map, but we're now at over 240 clinical research sites worldwide. And you can see most of that expansion has occurred domestically. We typically were based at the leading infectious disease departments in academic university hospitals. But you see here, we've expanded that domestically, primarily by reaching out to a lot of other government partners and network partners. We've brought in over 30 sites that are affiliated with Veterans Administration medical centers. We brought in additional commercial sites that are leaders in the ID field, and are really excited about engaging them. And they've been incredibly productive over the past couple months. We do a lot of operational support. This is an example of tracking the metrics of the average number of visits a day for one of our clinical trial sites that is enrolling 250 people in one study, highlighted in the gray colors, followed by, after they complete the second injection in these individuals, they're able to start the second study consecutively and in sequence. And you can see at some point here, they're going over 40 visits per day. And so the capacity, the square footage of these facilities has to be taken into consideration, as well as the critical post-vaccination monitoring for COVID itself. Of course, when people have a scratchy throat, they have a headache, fever, we need to test them for COVID. 9 out of 10 times, it's not going to be SARS-CoV-2 infection. So the throughput and active surveillance for COVID as well will continue for a year in some of these cases, up to 2 years for some of the platforms. And that, of course, requires the physical infrastructure at these clinical research sites. And some of them have utilized mobile clinical facilities, tents, and FEMA-like trailers that have been located in proximity to their clinics. This is the distribution of the co-VPN sites that we have domestically, and an overlay on the CDC ensemble model, which predicts out the level of COVID across the country. These models are available on the CDC website to see what the trajectory, what the likely trajectory is for SARS-CoV-2 infection in these areas. And you can see here they have, this was generated on October 22nd, and indeed portrays quite well some of the burden of disease that we've observed in the northern Midwest, particularly in Wisconsin and the Green Bay area, for example. Meanwhile, it persists with high levels of endemicity in the South, particularly Florida and Southern California. So just as a reminder, the typical development path for a drug or vaccine is through phase 1, 2, and phase 3 clinical trials, typically taking years to complete these. But for SARS-CoV-2, we're working as quickly as possible. We're not skipping any of these phases. As I mentioned, indeed, instead, we're overlapping them, stacking them on one on top of each other, but not moving into the critical phase 3 studies until we have sufficient confidence that there's good safety of the platform, as well as sufficient immunogenicity, immune responses elicited by the vaccine that are putatively protective against SARS-CoV-2 or COVID disease. And when we think about the development of these vaccines, we've also had to take into consideration some of the potential risks associated with vaccine development. And this is a summary by my colleague Barney Graham at NIH, talking about antibody-dependent enhancement or vaccine-associated exacerbation of respiratory disease. And this is a primary focus of attention because of some of the preclinical observations with SARS-1, and also because of some of the other viral infections that we know can be exacerbated by pre-existing vaccine immune responses, such as dengue. And dengue natural infection itself can predispose to a severe hemorrhagic fever upon subsequent infection by a different dengue virus strain. So this has been an ongoing concern. Fortunately, as of this date, We haven't seen any evidence of this in any of the clinical trials to date, which is very encouraging. A signal of efficacy that would elicit this concern would typically be observed in the first couple of months of these clinical trials. And indeed, none of the adverse events that have been reported from these clinical trials have at all been associated with these antibody-mediated enhancements of infection. But rather, these early phase clinical trials have been reviewed and published in peer-reviewed journals. On the left is the SARS-CoV-2 mRNA vaccine by Moderna, and on the right is the chimp adenovirus vector vaccine by Oxford and AstraZeneca. And in these following slides, I'm highlighting some unique differences between these platforms as reflected by the safety of the vaccines. The vaccines have been found to be generally safe and well tolerated, although there are some unique and interesting observations with regard to the frequency of some of the expected adverse events. On the left-hand side, the mRNA vaccine, as you can see after vaccination 1, elicits generally mild to moderate systemic adverse events such as arthralgias, fatigue, fever, fairly low levels of these. Some local symptoms such as erythema hit the severe level but resolved quickly. On the right of that column, you see vaccination 2 and a higher frequency of the moderate to severe adverse events after the second vaccination. Now, this is in contrast to the AstraZeneca chimpad viral vector on the right. In the columns under A, we see the first dose and the second dose. And in that column A, we see the local adverse experiences such as induration, itching, pain, tenderness. And you can see from the first dose to the second dose, for example, there's far fewer pain and tenderness observed. And similarly, in the bottom section B, these are the systemic adverse events collected from this early phase study. And for feverish or headache, you can see there being some mild and moderate and cases of this after the first dose, but after the second dose, again, fewer, both systemic and local adverse events observed. And this is a result of the different platforms used, the different strategies, the different ways these vaccines interact with the human immune system. And we'll be, of course, paying very close attention to these adverse events in these larger phase 3 trials as we move into tens of thousands of people who will be receiving the vaccines. And this is simply a summary of the immunogenicity of both the mRNA on the left, the adenoviral vector on the right. A lot of data essentially summarizing that they're both eliciting good antibody responses that are both binding antibody responses and neutralizing antibody responses. One of the critical questions that still is yet to really be fully answered, however, is the durability of these responses. And that will be answered later this year in early 2021. So this is the protocol pipeline for the CoVPN. The top 2 studies in gray are observational studies among individuals who have convalesced from infection, as well as individuals who have had acute infection. And we collect samples in these cohorts of individuals to understand their natural immune response to the virus. And then in the bottom 5 rows, we see Moderna, AstraZeneca, Janssen, Novavax, and Sanofi phase 3 clinical trials, with the enrollment periods highlighted in red and the follow-up periods highlighted in this yellow-orange. One of the things we will be paying a lot of attention to is making sure that among those individuals in these studies who do get infected, they get the best care possible. We don't want anyone falling so severely ill at home that it becomes a fatality. So we have observed that in the ICU, all had hypoxia and none due to shock alone. And in this series of 60 critically ill patients from China, again, all of them had hypoxia and none had shock on admission. So further, in a cohort of patients with early COVID admitted to an observation unit in South Korea, 12% experienced disease progression, and all of them based on hypoxia. So it's come to be known as these happy hypoxics that may be a feature of COVID infection. An example being a 74-year-old case without comorbidities who was asymptomatic but had an O2 sat of only 62%. And what is the unique pathophysiology behind this has yet to really fully come to light. But then this publication came out, which really prompted us to ensure that we had mobile pulse oximeters for those individuals who were diagnosed with COVID. They could take it home and collect their O2 sats while at home, because indeed, this O2 sat monitoring in patients who were discharged from an ER study observed that most patients who had an O2 sat of less than 92 after discharge experienced a very abrupt drop in their O2 sat rather than a gradual decline. And indeed, that has been observed in hospitalized patients as well, that when they decline precipitously. Another feature that the CoVPN has built is this voluntary screening registry. Again, I'd encourage you all to visit our site, preventcovid.org, volunteer now. It only takes 5 minutes and provides you a very good understanding of what type of characteristics, what type of data we use to collect in this registry to help us analyze who may be the best participants in these upcoming clinical trials. Because of course, someone who's retired at home, is living at home exclusively, has very little contact, very little probability of getting exposed, infected, and progressed disease, is not necessarily a good candidate for participating in the clinical trials. So the data we collect includes a number of things: demographics, health conditions, occupational activity, social activity, and then the registered users from each of the clinical trial sites use authentication to access their contact information that they can then reach out to identify these individuals. Within the first couple weeks of deploying this in July, we had almost 100,000 people register from around the country. And as of, I think this was 2 days ago, we had 456,000 people register. And I mentioned the risk score. We do apply a predictive analytics to the population who's registered, not only for hospitalization, intensive care, and fatality, but also developed a composite score that we updated that, again, synthesizes the likelihood of an individual to become infected and then to develop symptoms. And we developed this on an individual person level basis, and is currently being tested and deployed. And this is a snapshot of an example from the New Orleans Adolescent Trials Unit, who is participating in the Janssen clinical trial. They had over 1,400 people available to them. The recruiters then can filter on those 1,400, perhaps by race, ethnicity, and among those, 186 are reflected as Black or African American, Hispanic, or Latinx. And they can further filter them on this endpoint risk score to identify the optimal participants. We know that in Washington State, for example, over the past summer, there was an outbreak in the Yakima area associated with agricultural workers. So all of these factors are collected in the registry. This is a regional map of the catchment area in the Southeast, highlighting, the tremendous diversity that exists across that region. Excuse me. We have also developed risk maps to focus in on the sites themselves getting access to these risk maps. We have a Veterans Administration medical facility, for example, in Jacksonville. Of course, the recruiters and the people in Jacksonville know their community best, but this highlights some of the race ethnicity disparities as well. When looking on the left, the darker color is the pre-existing morbidities, and on the right is the race ethnicities, in this case, a predominance of Black African Americans in those neighborhood zip codes with the highest level of pre-existing morbidities. Similarly, we've done this in communities where we've also looked at different pharmacies which I think all of you would be interested in. We look at priority pharmacies that may exist in these areas of high risk with high density pre-existing morbidities. We're tracking these, working with the local sites, and providing them lists, actual tabulations, enumeration of these vaccines that, for example, we would recommend them to be reaching out to for potential sources of recruitment. We also have a tremendous amount of other materials. We developed a fantastic public service announcement narrated by Harrison Ford that I could show you if there's time. And this material is really trying to address a lot of what we've been feeling in this epidemic, a lot of the uncertainty of when this will end, of when we can gather as we've been accustomed to, and how best to encourage people to step up and participate in these clinical trials. So with that, we have learned an immense amount, but of course the big learnings are yet to come, and that is the true efficacy of these vaccines. But as we have predicted over the past couple decades, global pandemics will result from trans-species transmission of virus pathogens. They have occurred in the past and will continue to do so. Advanced technology makes opportunities for very rapid vaccine development, and advances in therapeutic options has contributed to a reduction in morbidity and mortality since the beginning of the epidemic, which is another very strong rationale for trying to at least flatten the curve, as was previously highlighted earlier in the epidemic, as a strategy for extending this and reducing the morbidity mortality such that perhaps we'll have better interventions in the future, and that has turned out to be the case. We've learned, of course, transparency and scientific evidence should be guiding tenets to public health policy. The community and public trust demand a coordinated effort. And this pandemic has unfortunately highlighted some of the weaknesses in our current healthcare system that will require immediate and lasting changes. And ultimately, effective vaccines may be the most efficient and effective means in controlling the pandemic. I work with just an incredible group of people, hundreds here in Seattle, thousands in the country, tens of thousands totally around the world in this concerted effort. And I'd like to thank you for your attention.
Thank you, Jim. Thank you, Dr. Kublin, for that awesome presentation. I invite individuals, we have plenty of time for questions. This is just a great treat to be able to ask you kind of what we've been dying to know about research and these vaccines. So thank you for spending your time with us. The first question comes from Steve Sachs. Steve, I'm going to unmute you so you can ask your question if that's okay with you.
Yeah, hi, Steve.
Go ahead, Steve. Oh, probably caught him off guard by unmuting him versus just reading his question. As you review for the durability of protection from the different vaccines, do you expect there will be a difference in durability between the different vaccines? And if so, will that create issues in trying to prioritize who gets which vaccines?
Oh, that's a great question. So I do predict that there will be some differences in the durability of these vaccines, and that indeed the Janssen clinical trial using the Ad26 vector is testing just a single dose of the vaccine in the current study that we're working on. But we're also in discussions with them and talking about a 2-dose regimen. And so what we're going to see is another study launched sometime in the next few weeks by Janssen that will be evaluating this 2-dose regimen. And so we'll be able to compare very directly the immunogenicity and durability of the immune responses from this Ad26 vector for 1 dose and 2 doses. Of course, I'd predict the durability will be greater with the 2-dose vaccine regimen, but it may not be necessary. I mean, we just don't yet know what that level, what the critical threshold of either binding antibodies or neutralizing antibodies will be for the efficacy of these vaccines. And that brings me to the correlates of protection, which will be such a tremendous focus of attention by the computational and statistical folks to help us identify what is that critical threshold of an immune response above which you have a high likelihood of protection and below which you may need either a booster vaccine or an annual vaccine. We don't know the answers to these questions. I think we could also hypothesize what populations may be most suitable for a less durable vaccine, but may require boosters annually. So all of these implementation and policy issues around the deployment of an effective vaccine is not directly our responsibility. That really will be under the auspices of the CDC. And we'll wait and see. The National Academy of Medicine did come out with a fantastic recommendation of how to deploy these effective vaccines, and it's available free online.
Thank you so much for that answer. And one, I'm not sure if you have insight on this. I do know these resources are up and available. Which vaccines will have more kind of standard refrigeration or at least standard freezer temperature storage?
Yeah, that's also a great question, and we're in the process of compiling, the storage requirements of the phase 3 material and what the developers are working on for the deployment of the final biological product and the stability of that product at 2 to 8 degrees. So we don't think any of these, unfortunately, will be lyophilized. The lyophilization process is just not built to scale to accompany that. So there will be cold storage required. Hopefully the stability and storage, at least when it hits the clinic, can be at 2 to 8 degrees. But that has not been confirmed yet. Some of them still are requiring as low as -80. So that's not something that's very sustainable. And we're confident that, that they'll be able to get to -20, if not 2 to 4 degrees.
And is that even of the Pfizer vaccine eventually, you hope?
We hope so. Yeah, that
Fingers crossed.
Fingers crossed. Yeah, exactly.
And then, yeah, you were talking about the 2-dose series. And I know that from what I've heard is that the 2-dose series is either 21 or 28 days apart for the minimum interval, correct?
That's correct. Pfizer was using a 21-day. The Ad26 viral vector 2-dose regimen will be a month apart. The Moderna study was 28 days. And AstraZeneca, I believe, is also 28 days. Most of and the protein adjuvant vaccines by Sanofi and Novavax will also likely be 1 month.
And I think, with your experience in vaccinology, can you speak to a minimum interval versus kind of a maximum interval, or if it has to be given exactly 21 or 28 days apart?
Sure. Yeah, there are certainly windows in those vaccination. I think what we don't want to do is give it too early. So I'd say 21 days being a minimum. And for most of these vaccines that do require 2 doses, I think they'll likely recommend it's at least a month. The boost of the response is oftentimes more robust, and the durability may be better if there's a little bit more of an interval, 4 to 6 weeks after the prime. But of course, we're trying to refine that and do those studies while moving at warp speed is quite is not necessarily the top priority. First and foremost, we want to really identify what the probability of success for efficacy is with these standard vaccine regimens. of course, the correlates, this highlights also the tremendous importance of the correlates of protection, because it's these correlates of protection that could really help inform what may be an optimal regimen for prime boost.
Not an easy question when you have competing priorities to weigh in on. Megan, you had a question for Dr. Kublin.
Yeah, thanks for joining us and sharing all of your knowledge and insight, Dr. Kublin. I was just curious, and maybe it's like too soon to tell, but is there going to be sort of a reverse learning opportunity with this? So you talked about how you took learnings from HIV vaccines and other things to inform this one. So what can you take from this experience and maybe apply back to other developments or refinements of existing vaccines?
Yeah, that's a really exciting opportunity and a great question. I haven't, took the time to reflect on that much, not being able to apply it back yet, but I'm very excited to do so. I think, the advances in the nucleic acid platforms has been really exciting. I think we always wanted to test these mRNA vaccines with HIV envelope proteins, and now we do have a commitment and even a lot reserved for the manufacturing of an mRNA HIV envelope protein. And we'll compare the immune responses elicited by that nucleic acid delivered protein compared to the recombinant standard soluble HIV envelope protein that's manufactured beforehand. So, that already is something that has been a springboard from these activities just in the past 8 to 10 months. And there surely will be others. We'll be looking at different adjuvants with these vaccines. We're hoping that these adjuvants will improve the durability of the immune responses. And then the tremendous knowledge that we're gaining from these viral vectors that are inducing these CD8 T cells, and how that may provide some additional protection, perhaps to some of the severe disease that's associated with SARS-CoV-2 infection. And how to apply that to other pathogens, whether it be HIV, malaria, tuberculosis, some of these global health infectious disease pathogens that are still killing. TB is expected to have 1.5 million people perish this year from TB alone. So some of these other infectious diseases remain very significant severe public health problems. Great question.
Thank you. Are there other questions? I'm looking for raised hands or for people to type out questions for Dr. Kublin. We did have a question about, does the vaccination, will that complicate testing for the vaccine or for the virus in patients, or immunity in patients, or past exposure for that? Has there been any exploration into that?
Yeah, that's a great idea. So the vaccine-induced responses, may result in a vaccine-induced positive test, although most tests for active infection, of course, are relying on a nucleic acid PCR test for detection of the SARS-CoV-2 virus itself. We're hoping that many of the routine tests under development are including some of the core proteins that are not included in these vaccines. So that's one benefit. And then, in sero prevalence studies in which they're looking at antibodies against the virus, that's another situation in which vaccinees could be misinterpreted as having previous infection, whereas they're simply eliciting a good response to the vaccine. So if I understood the question correctly, I think these are likely to be an issue for us. We of course include that in the consent form, and highlights the importance of people getting the appropriate diagnostic when necessary.
Thank you. I do think that answers their question well. Another question that I think really is on people's minds, and we've heard discussion of, is COVID-19 going to be the new flu? Do we see putting the COVID-19 vaccine in the flu vaccine in the future? Is that even feasible? What are some of the discussion points you see in looking into the future with the future of COVID vaccine and or COVID infection, and then possibly with the vaccine as well?
Yeah, I mean, if we're effective in getting a vaccine, and as I mentioned, there's a high probability of that success, and it's deployed predominantly initially in the adult population, we would expect that the majority of adults to be protected, and then we'll drop down into the younger ages when we've confirmed that the vaccines are safe and that there's some level of efficacy. We'd want to include them in childhood vaccinations, but I suspect that will take some time. And so it could be a virus that circulates predominantly among children. Fortunately, children have very low rates of severe disease, but I think, as you've probably heard, there are some immune-mediated inflammatory reactions to this infection among children that can be quite devastating. And so, they will benefit from a vaccine as well. I think in large part it depends on the durability of the vaccine-induced responses that are protective, and then the variety or variation and recombination of the virus, and to what extent the virus becomes resistant to the vaccine-induced protective responses. And that is simply something that we're going to have to follow very carefully. It's also a great question.
Thank you. A lot to look down the road to. Well, I don't see any other questions at this moment. I have a feeling there's a lot more questions up people's sleeves, though. But thank you so much for sharing your insight with us today and your time, and furthermore for all the excellent work that you're doing at the Hutch, both on developing vaccines for COVID-19, but all of the other critical illnesses of the time. And I think that we all appreciate having such a great resource in the Fred Hutch being in our own backyards. So thank you so much for participating, and then also all of your work you're doing. Thank you so much.
Thank you, Jenny, and thanks for the other attendees for your attention, and best wishes in your meeting.
Thank you.
Okay.